Thank you for joining the U.S. Army Corps of Engineers Northwestern Division for today's conference call covering test flows from Fort Peck Dam. This call is open to the public to provide information on the status of the test flows to ensure compliance with the 2018 biological opinion. I am Eileen Williamson, and I will be your moderator. This call is recorded and placed on the Defense Video and Imagery Distribution Service for reuse at www.dvidshub.net/unit/USACE-NWD. Your participation acknowledges your consent to be recorded. All lines have been placed on mute. When we get to the question and answer session, to unmute your line, press *6 or use the microphone in the WebEx application. Do not place the call on hold, but you may need to use your phone's own mute function. The question and answer session will be at the end of the call. John Remus, Chief of Missouri River Basin Water Management, will provide an update on releases and test flows from Fort Peck. Joe Bono, Manager of the Missouri River Recovery Program, will provide an update on monitoring. And Pat Broughton with the U.S. Geological Survey will provide an update on the fish. With that, I will hand the call over to John Remus.
Very good morning, or good afternoon, everyone. Can you move the slides forward?
Forward like?
I don't see the slides where I was going to talk about.
Oh,
the next slide.
I don't have any slides from you, John.
Okay. Well, 1 second then.
You send them real quick. I can put them up.
No, I'll just talk to them here.
Okay.
Okay. We are in, as many of you may have known, we delayed the second peak about a week due to high stages at Williston, North Dakota. And because we were about 2,500 CFS below our planned retention flows, the new peak was set at 20,000 cubic feet per second instead of 22,500. We reached that peak around the 26th of June, held the peak till about the 29th. Came down 29th through the 5th, 29th of June through the 5th of July to about 10,000 CFS. Then on the 7th of July, we dropped to about 8,000 cubic feet per second. That's where we're at today. We'll be holding that flow, 8,000 CFS, average daily release, or average daily flow at Wolf Point through the end of August. And it's August or around September 1st, we'll resume normal operations. That's the water management side of it. From the communication side, that we holding these weekly stakeholder meetings every Wednesday afternoon. And we'll continue those at least for another couple of weeks, I think, until we can see how the 8,000 CFS is working out as we go down the system. We'll continue to have our monthly stakeholder calls, which are wider basin, or complete basin calls. Our next one is actually tomorrow afternoon at 1 o'clock in the afternoon. Eileen, I think, sent out something, or will send out a meeting notice on that. And then, of course, we have the feedback loops where you can either provide stuff through email to our Missouri River Water Management email address, which is on one of the slides, or you can submit it through the HydroViz tool, which is also the link to that is also on the slide. With that, I think I'll turn it over to Joe for anything that he may have to add here at this time.
John, over to you, Joe.
All right, thanks. So we don't have new updates, physical monitoring side. If you've been on any of the last couple of calls, there's nothing new to report, but I'll go over it for folks that maybe haven't been on the last call or two, just so you know what's been done and what's coming. So we've conducted aerial photography, LiDAR surveys before the test flow started. That was completed. During the first peak of the test flow, that was completed. And what remains is the post-test flow work, the photography and LiDAR, that's to come. From the physical pump site surveys, there were surveys conducted before the test flow started. That was complete. Surveys conducted both during the first peak and the second peak. Those were completed. And what remains is the post-test flow survey work. From the cultural resource monitoring standpoint, there was monitoring done during the test flow. That field collection is complete, and there's another round similar monitoring test flow work remains. And many of these that will receive data in the fall sometime and reports fall, winter, depending on which data we're talking about. So as we receive results, and we'll share results with you, but that's in the future, quite a ways still. Dam safety monitoring, there was pre-test flow inspection. It was completed. Then the slab and wall monitoring that has been ongoing during the test flow. So that happened all the way through. The spillway flows are now ended, but so that dam safety monitoring work is largely completed. The water quality monitoring is ongoing. It started before the test flow began, and it continues and will continue through the summer. So that's, again, not new. You've heard that before if you've been on the last couple of calls, but that's a summary of the non-fish type monitoring that we have going on, what's been done and what's still to come.
Thanks
So
a
that's
lot,
all.
Joe.
Yep, thank you.
We'll hand it over to Pat.
Thanks, Eileen. Okay, good afternoon, everybody. So in today's presentation, I'm just going to give you a review on the status of the reproductive females in the upper Missouri River response reaches. And then I will touch on some new information for you that stems from a larval drift study that accompanied the whole Fort Peck flow test scenario. So I know some of you have been on this call since inception. There may be some new people on, so I'll just run through this real quickly, bring everybody up to speed. On April 29th, or April 26th, excuse me, the first flow pulse was initiated, and that peaked out around 18,000 CFS at Wolf Point, and this is the black line and the left axis on your screen. Those flows came down, entered the period of retention flows that lasted for about a month. And then on June 17th, flows were increased again for the second peak, projected to stimulate spawning, aggregation of fish. And then that peaked out about 20,000 CFS at Wolf Point. And then, as John had mentioned just a little bit ago, flows have decreased, and now we're right around 8,000 CFS. Also, for those of you who have been on this call for the past several weeks, you're familiar with the females. I'll just give you a quick rundown, review of these females. There were 5 females originally in the response reach. 2 of them were the wild females, code 262, which is the gray bar that you will see. The other wild female was code 272. These fish entered the Missouri River response reach in early to mid-May. They stayed for a little bit, and then they exited during the retention flow time frame, late May through early June. Code 262 did get as high as about river mile 1650. Code 272 in the brown got up to about 1620, 1630 in that Culbertson area. These fish entered the Yellowstone River, and based on captures on June 24th and 25th, these fish were known to have spawned, most likely in the Yellowstone River, because after they left the Missouri River, they were in that system with a lot of males in the lower part of the river. Now I'll jump to the hatchery origin females, codes 422, 337, and 342. These fish were collected just last week on July 2nd through 3rd. All three of them were still carrying eggs, so they had not spawned as of that point yet. So we are going to, being that these fish were just handled last week, we're going to give them a couple more weeks to see if they do spawn. You know, the water temperatures right now are starting to increase with the very welcome heat that we've had up here. So we will see in a couple weeks, handle these fish again and determine if they have spawned. Okay, next slide. So staying on top of the reproductive fish, doing the tracking and assessments is only one part of the whole flow modification project. There was a second portion to this project, survival, drift, and dispersal of paddle sturgeon free embryos, very complementary to the whole spillway release. The objectives are really to evaluate the existing drift and dispersal models that exist for the Missouri River right now. There are a variety of models out there that assess how far, how fast free embryos will drift and disperse in the Missouri River system. Some are from field studies, some are from lab studies. So those studies do provide a very good backdrop for what's happening or what could potentially happen in the river. And really this work is looking at addressing that transition. How far will these free embryos drift before they transition to a settled larvae, getting out of the water column and living on the river bottom. Also, as part of this project, you could have, one or two wild spawners in the system. And even though the free embryos could hatch, there aren't a lot of them that end up coming out of an incubation period. So by stocking a large number of free embryos produced from hatchery broodstock, those free embryos do provide a large sample size of fish drifting down the river from which we can sample and draw inferences on drift and dispersal. As part of this, we were able to assess the initial survival of these one-day post-hatch free embryos as they were released in the warmer waters of the spillway and as they moved downstream and gradually mixed with the colder powerhouse releases. We had two sites. These are about 4 and 8 miles downstream, but we also did tackle one additional site further downstream, about 170 miles in the Missouri River, just upstream from the Yellowstone River confluence. And based on travel time of the fish to these first couple sites, and based on some other work we've done in the Missouri River, you can kind of get a handle on how far and how fast these fish are going, and target a specific portion of that dispersing population in the lower portions of the river. And that's what we did, hopefully to get at some of the longer term survival for these fish. So on the right side of the picture, you have the spillway flowing. The study was conducted on June 26th. I think we were on the first day after the peak flows. So you had the warmer water coming down the spillway, a little more turbid as well. And if you look at the picture on the left, you see one boat stationed at the outfall of the spillway where it enters the main channel. And you can see the little turbidity plume coming from the spillway in that outfall area. Okay, next slide. So projects such as this takes a lot of people to make it work. First of all, it starts in the hatcheries. There was a very large supply of hatchery broodstock down at Gavin's Point Dam National Fish Hatchery. Down there, the hatchery experts spawned out multiple male and female pallid sturgeon, incubated those eggs for a couple of days. They were subsequently transferred up to the Miles City State Fish Hatchery, where incubation was completed, and they were used as a source for the drift study. So on the upper left, you have a picture of the fish in the hatchery tanks. On the upper right, you have all these fish that are being bagged and ready for transport to the spillway release site. And in total, we had right around 350,000 free embryos for the drift study. So when we got to the spillway channel, We acclimated these fish for about 30 minutes to match the water temperatures. So they were released right at about 16.5 degrees Celsius. They were released on June 26th, right around 3 o'clock in the afternoon. Okay, next slide. So as I mentioned earlier, we had 2 primary sites to assess that initial survival. The yellow dot right at that spillway outfall there is the release site. Site 1 was about 4 miles downstream. Site 2 was about 8 miles downstream. And the study just happened, a couple weeks ago, so we're still working through getting all the numbers crunched and verified. But there are roughly 1,000 free embryos that were collected between these 2 initial sites. I can tell you that when we pulled those free embryos up from nets, dumped them in the pans, they were still swimming around alive and kicking. So initial survival did happen. And also 170 miles downstream, we had a site for a couple of days that we targeted. There were several free embryos collected at that site as well. However, given that as you drift or you get further downstream, you have contributions from shovelnose sturgeon, paddlefish free embryos as well. So we'll have to conduct genetic analysis to differentiate those free embryos from further downstream as pallid sturgeon, paddlefish, or shovelnose sturgeon. And that will do it.
Great, thank you, Pat. If there are any questions, feel free to go ahead and raise your hand using the app or type your question into the chat. Not seeing some of our regulars. We'll just go in alphabetical order then by state, starting with Montana. *6 to come off mute or use the microphone to unmute and ask your question. Okay, moving from Montana to North Dakota. And then alphabetical order, state of Iowa. Kansas. Missouri. Nebraska. South Dakota. And Wyoming. Okay. John, I will hand it back to you for closing comments.
Well, thank you again for everybody for tuning in today. We will, even though we're down to a steady flow here, we will conduct another call next week and probably the week after. And then we'll see whether or not we feel we need to keep these going on. So again, everybody have a good day, and we'll talk to everybody next week. Bye.
Thank you. That concludes today's call.